Hi, this is Terry Kushma here at Penn State University. And what we're doing today is looking at materials and nanotechnology. Specifically, we would like to look at nanoparticles for medical applications. This is a probably 4 lectures long, so this will be part one in that series. So as an introduction, nanomanufacturing supplies many unique materials and processes for biological applications. These might range from nanoparticles to drug delivery to prosthetic devices. And what we're going to key in this one is really the look at the cellular level and understand biology for a background and how we would apply nanomaterials to that application. We're not covering prosthetic devices within this presentation. So it's necessary to understand how biological entities like cells interact with products crafted on a nanoscale. So this packet will review basic biology with emphasis on scale and interface with materials. Then we'll look at common nanomanufactured biomaterials with associated applications. So what we need to do is review first the boundaries of this application. So we'll overview the basic cell structure. Next, we'll look at cellular function, particularly what DNA does in the body and the importance of DNA and its relationship to proteins. We'll also look at the cell itself and view other structures in vivo. So basically, we need to appreciate the cell interfacing with nanomaterials or materials interfacing on the nanoscale. So this overview will be on cancer, specifically how we can use nanomaterials to defeat cancer. So narrowing it down, there's a broad range, I guess, lifetimes of material we could present in this area of nanomaterials for medical research. But what we want to do is specifically look at materials that are state of the art. I mean, they're not new, but they're within the last 20 years, and these are well-established drugs, and they actually show the relationship and what we need to do with future drugs. So I thought it was a good interface for a presentation. So what we're going to do first is, this is our outline for the next few lectures. We're going to look at biocompatibility, and then we'll do a quick overview of cellular interaction. We'll talk about the scale, the size, and look at a generic animal cell and see how, an understanding of that, we can apply an understanding how materials interact with that generic cell. Then we'll look at materials for biological interaction. And the material that we're going to look at the most in this presentation is called a liposome. And what a liposome is it's a biomolecule. So it's a man-made nanoparticle, and we're going to use that for cancer treatment. So that's a current cancer treatment. So an understanding of that material leads a long way into looking at this specific subject. Then we'll also look at metal nanoparticles, specifically gold nanoparticles and silver nanoparticles. And then we're going to look at another system called nanoshells, and then some examples of bio-nano applications which are common with nanoparticles. So let's define, and a good starting place is, what is biocompatibility? So biocompatibility is the ability of a material to perform with an appropriate host response to a specific application. So for example, sometimes we would want to have a material act and the body to know that the material's in your body, the new material or nano coating. So we can look at, in particular, say like a screw that they would use to mend your bone. So if you had a catastrophic break in your femur or something, they might take a screw with nano coatings on there and then screw your bone together. And basically what you want the body to do is react to that screw and form bone around that screw. So you don't want it to ignore it. You actually want it to see it, and you want the screw to then be incorporated in with your natural bone cells and grow bone cells over it. As a corollary, we could have the exact opposite. We might have a material like a stent or a catheter that you insert into your body, and we probably don't want the body to really, in fact, see it or to react to it or start growing cells on it at all. So in that case, we want it to be inactive in the body. So biocompatibility is interesting that it means both inactivity in the body or activity within the body. But importantly, we know what we want that material to do, And atomically, with nanotechnology, we design how that material interacts in the body, whether the body reacts or doesn't react to that particular material that we insert. So to understand biocompatibility, the nanotechnologist must amalgamate an understanding of materials and biological response. The first part of this package focuses on cellular function. This allows us to appreciate the biological scale and cellular activity. The second part of this packet examines biological response to engineered material at the nanoscale. So biological or biocompatibility, the biological response to an engineered material should consider both the long and short-term response. So the acute response is the near-term reaction of the body to the material, and the long-term response could be chemical release, chemical degradation, and shedding of particles, etc. And interestingly, materials, I mean, probably a typical one is cobalt. A lot of times they could use that for a bulk material for things like implants, like a knee implant or a hip implant. And people aren't allergic to people. They test people for the allergy to cobalt. They're not allergic to it. And then a number of years, the body comes up with an allergy to this material. So that would be considered the long-term response. So sometimes over time, the body will react differently. So this is important for those devices like dental implants, prosthetic implants like knees and hips, etc. So these things must be considered. Sometimes the acute response, is how the system would heal. So sometimes we want to elicit the healing response from the body, so we'll engineer that into the material. So let's take a quick overview of cellular interaction and look at the scale and the size of a generic animal cell. And again, this is quick, I guess, in time. I would encourage everybody to take a class on things like molecular cell biology, and that unto itself would be, per se, a 3 or 6 credit course in college, and you would certainly need even a bio background. So we're going to go over this in a relative short amount of time. And again, this is an overview so that we would be able to get a foundation of how we would apply the material. So again, I think that you should try to look outside of this lecture if you're interested in this material, or we could explore it further in a different class. But in this class, we only have time to do a short look. But I think it's a nice short look to give us the boundary conditions of how we have to design our nanomaterial. So the cellular size, the logistics of carrying out metabolism sets the limit on size range of the cells. As an object of a particular shape increases in size, its volume grows proportionally more rapidly than its surface. So for eukaryotic cells, this is the cell that we're going to look at the most because it's in mammals, etc. There's proeukaryotic cells, and they're like a mold slime that's in creeks or whatever. We're not going to look at that, but we're going to look at cells that have a nucleus. For the most part, they're around 5 to 100 microns in diameter, and of course this size and shape depends on their function. Generically, I would say, just for an idea of a general scale, well, how big is a cell because of that range? I'd say maybe something to have in mind is a generic cell is 10 microns in diameter. A red blood cell, for example, is 7.5 microns in diameter, and we all know that it's shaped kind of like a frisbee, like a platelet. So that's the large diameter, and it's thinner, and naturally it's bending and folding, etc. So these relative sizes are per se a conceptual idea anyhow, but I would propose that we want to keep in mind that a cell might be 10 microns in diameter, and then we have to look at other portions of the cell, what's included in the cell, as far as like the mitochondria or DNA, etc. So we want to take a look at those sizes and scales too. So why are cells microscopic? So for objects of the same shape, the smaller the object, the greater its surface area to volume ratio. So interestingly, this is certainly a definition that we talk about all the time in nanotechnology, because this is the definition of a nanoparticle, that it has a high surface area per unit volume. So if we were a corollary to look at this as an example, if cells were larger, The rates of chemical exchange with the extracellular environment might be inadequate to maintain the cell due to a greater distance between the cell membrane and the nucleus. So basically this says that for the most part, the functionality of the cell, its goals to maintain life or to build life within an organism, sets its size. That's the boundary value. And what would be efficient for these cells is communication on the level of 10 microns or so. So cells that are like that, they're able to interface with other cells, and that interface builds up a system, and that system would be an organism. So why are cells microscopic? Again, by dividing cells into large cells into smaller cells, the surface area to volume ratio is maximized. This serves the cell's need for acquiring nutrients and expelling waste products, and actually communicating via proteins and excreting and taking in proteins. So this relationship explains why larger organisms don't have larger cells, but they have more of those cells. So how does this standardization of cell size impact us in nanotechnology as engineers and scientists? Well, the universal size for cells means universal scaling, and this dictates the manufacturing tool set. So the same design algorithms that we would use to understand cells in mice, we could use to understand cells in elephants or in corn. So this is important because this technology is vast, but the vastness of this technology actually cures a lot of issues, and we'll look at a lot of different types of cells. So techniques and applications can be shared across the nanobio market. So for example, if we were to make a cancer drug and design a cancer drug, which we'll look at today, the liposome, certainly we could use that in mice, and we can also apply that directly to horses or people. So that's a good application, and shows us why this is a nice goal, or it's fortunate that nature made cells all the same size, because now we can use products across a large variety of applications. So this shows us the cell size versus surface, and as we divide larger surfaces into smaller units, we get a greater surface to volume ratio. So we've seen this a number of times in our discussions of nanotechnology. And this is a nice slide to show us some standards to give us an idea of what we're discussing. A gas molecule being about 10 to 1 nanometer, a virus being 2 to 100 nanometers. And that's interesting because the size of a virus is now, the size of the technology they would use to create gates on transistors for cell phones. So transistor technologies in the area of viruses. Tobacco smoke, really huge. 10 to 300 nanometers, and we could see that things like smoke and dust, etc., dangerous for the clean room, right, and yield. And bacteria is huge. Bacteria is 0.2 to 10 microns, and certainly that is in the area where we could use nanotechnology to help with bacteria, control bacteria, abate bacteria, etc. Fog, rather large. And then a red blood cell, I always look at that as a milestone of size, 7.5 microns. Dust, pollen. A human hair is about, I always say, about 100 microns, because it's a nice even number. And I think that's a nice thing there, 40 microns. So people say 30 to 40 microns is what a person could see with their eyeball. So when you're, and you're young, in your 20s or something, 30s, before your vision starts going bad in the 40s, 50s, you could probably see dust particles, thread a needle and stuff like that, down to about 30 microns. So really being 1 micron is absolutely tiny, well beyond the visual acuity of the human eye. So interestingly, the cells generally accept particles that are 100 nanometers or less. So that gives us the ability to create things on a nanoscale, because that's actually like the physical currency that cells respect and they're able to transfer or take in. So larger particles and non-spherical particles are really not accepted by cells. So it's interesting that people have done studies on cells, and they have done things like taken nanorods and nanospheres and try to have cells uptake those, and they'll always preferential to the spheres other than the rods or a triangle or a square, etc. So the thing that the cells like the most is spheres and circles. So nanoparticles almost must also elicit familiarity to the cell. So coatings such as proteins and sugars greatly add to this acceptance. So if we wanted to do something like have a gold nanoparticle be able to breach the cell membrane, it would recognize gold as a metal and wouldn't like that. So what would happen to the gold is there has to be a coating or a skin on there that would be some type of protein, sugar, polymer, and sometimes the body naturally coats that in the blood system. You can acquire these things will actually attach to gold, or you can make them in a man-made fashion. We'll actually look at those issues later at the final portion of this presentation. We'll look at coatings on metal nanoparticles and cell acceptance of those materials. So we also will look at be looking at cells and ligands to develop engineered medicines, and later we'll introduce the liposome, which is a man-made biological nanoparticle that's widely used in medicine today. So the liposome is incorporated into many parts of this discussion and the presentation. So for example, we're going to look at DNA and its importance in proteins, etc. But we look at DNA for cell division. And what's necessary when a cell divides is that the DNA divides. And part of the liposome cancer treatment is that it prevents the DNA from dividing. And if you don't have cells divide, then they won't procreate, and that's an issue with cancer. So cancer is known as a rapidly dividing cellular network. What we're going to try to do to cure cancer per se, or to alleviate the cancer, is we'll keep the cancer from rapidly procreating. So this is a generic animal cell, from the NSF, and it's not any particular cell. It just shows the items that we're interested in the cell itself. So I don't know where to start. It's so dynamic and so many things to see in here, but we can start maybe looking at the cell membrane. And what the cell membrane is made up of, interestingly, is it's a phospholipid, and it's a phospholipid biwall. So what a phospholipid is, it actually has a water loving head and a fatty acid tail. So one portion of this biomolecule is going to point towards water, and one portion is going to point away from water. So with cell walls and some cell membranes, more appropriately, in animal cell membranes, cell walls are in plants. Cell membranes, what they're made of is this bilayer of a phospholipid. Interestingly, the nucleus that encapsulates the DNA, and the DNA is important because it actually makes proteins, and proteins carry out life functions. So to protect the DNA from non-ideal conditions, it's actually housed in the nucleus within the cell as another protective layer so that it doesn't get damaged. So what the nucleus does, it's also made, this nuclear envelope is also made out of a phosphid bilayer. So that's interesting. And what we're going to do is take that, same thing that nature does. We're going to copy nature, and we're going to use the phospholipid bilayer as the cellular wall to create our liposomes for drugs later. So we're actually emulating nature, which makes a lot of sense. And one of the one sense is we kind of want to do what nature wants to be done. So nature is used to looking at lipids, lipid bilayers. So we're going to design drugs that are that the body's not going to react violently to per se, and see as foreign objects. And as we can see in this picture, we have DNA. And DNA is enclosed in the nucleus. And what DNA does is, DNA is like the hard drive within the body, and it has all the programs and all the cellular function. And it's interesting that the cell, like in your eyeball, has the same DNA information as a cell in your toe. But your eyeball does different jobs as your toe does, or your heart, or your liver. So the DNA has all the functionality on it. And what happens is the body only expresses a portion of that DNA to do that particular cell's function within the body. And what it does is, we can say, is like an analogy, it would call out from the master program, which is the DNA, it would call it a sub-program, which we're going to refer to as RNA. So RNA is a subset copy of the main memory, the DNA unit within the cell. And what that RNA is going to do is it's exported outside of the nucleus into sometimes the endoplasmic reticulum within the cell, et cetera. And we're going to have a helper entity called the ribosome. And what that's going to do is take a number of amino acids according to the data information that's incorporated on the RNA. The ribosome is going to ride along the DNA, and it's going to have chemical information on the RNA to assemble amino acids. When these amino acids are assembled and folded, what we're going to do is get a functional protein. And these functional proteins are going to be the keys to releasing energy and making things happen in the body. So the biology guys often say that, that the body operates, or life's functionality is due to proteins. And naturally, we can see the cascade of events here and why you don't want things like radiation damage, because they could damage your DNA. Once your DNA is damaged, then your RNA is damaged. Your RNA is the encoding for the protein, and if the encoding is wrong on your RNA, then your protein would be incorrect. If your protein is not folded correctly, then it's not going to carry out a specific function in your body, like digestive function or etc. So if your protein is suspect or bad or not created in the right way because your DNA was damaged, the RNA is damaged, then your protein is going to be damaged and not carry out the functionality that it's intended to do. So form fits function. From the previous slide of the animal cell is a generic representation of a cell. Really no cell looks like that, but it gives us a good idea to discuss the relative applications of these components within the cell. So cells themselves are extremely diverse in appearance. They're modified for a specific purpose, and this purpose lets them express a certain functionality in the body. So different cells contain a different amount of organelles depending on the role they play with the body. The form fits the function, and we select materials to interact with that cell function. Again, we'll reference the cell function and then relate this information with cells. So that's the goal of this, at least certainly the first part of this presentation, is to discuss cellular function on a moderately high level so that we could actually interface nanomaterials to the cell. So this is some cell diversity that we can take a look at. So these are the different cells that were within the body, and certainly they're all not 10 microns. They could be of different sizes, but we have things like neurons, which could be in your brains, etc., and what they provide in your body. We could have connective tissue. We could have blood cells, bone cells, skeletal muscles, and cellular linings, epithelial cells. So the epithelial cells are important because they give like structure to the body, and we'll learn later on that the epithelial cells often aren't arranged appropriately in a cancer region. So cancer itself is not a normal part of the body, and it exhibits defective epithelial cells. And we're going to take advantage of that with our liposome drug, so that difference, we're going to identify that difference, and then we're going to create our nanoparticle to interact with that difference to be a cancer cure. So you can see, our objective within this presentation. So let's examine cell structure and related function. Some key points are the roles of proteins, the role of DNA, and the cell communication and interaction with materials. So as I said before, the cells carry out and communicate through protein interaction. That's a very big, I guess, macro idea. And the DNA makes a template to RNA, and then the RNA is used to create these specific proteins. So that's really the game function, and we want to look at DNA in that role. But we also want to look at, simply when we look at cellular division later in the presentation, that the DNA actually has to unzip itself, and there would be like the primary and secondary DNA, and it would per se clone itself. What we're going to do with our liposome drug is we're going to prevent that DNA from splitting. Therefore, we can't have cellular division, and that's going to like abate the cancer in the body. So we have to take a look at the cell wall and see how that works. We're going to look at the cell wall itself and see that it's made of a self-assembling material called a phospholipid. So again, this is a good lesson in nanotechnology because we're looking at self-assembly and seeing how nature uses these low energy regimes efficiently to create biological entities. So phospholipids are always used as a base to create liposomes that were used for drug delivery systems, so we'll discuss that later. The cell wall, or cell membrane more appropriately, is made out of a phospholipid sheet that has ports that regulate nutrients, waste material communication, and interaction with other cells. So basically, this cell wall sees, communicates, and carries out life functions, or provides a conduit to do those activities. And proteins are the key that opens and closes these ports within the cell, and man-made materials must interact and communicate with these ports and these proteins, or use these proteins within the nanoproduct. So this shows a membrane structure, a generic membrane structure, and this is the lipid biwall that we can see in the middle, represented by those yellow tails, and those purplish spheres are the hydrophilic phosphate heads. So we can see that this is a lipid biwall, that we would actually have a lipid facing, in this picture, up and down or north and south. And basically what they do is form a wall. But we have these other issues shown in the brown. Those would be ports where we can communicate with the extracellular matrix with outside of the cell. So those are like openings, like doors. And what was shown in the green, those like hair-like substances, they might interact with proteins, and what they would do is they're like the lock or the key. Protein would come over as a lock and then unlock those doors so that we could pass things in between the cells. And the things we would be passing are most often like proteins, like hormones. So hormones are an example of a protein, and these hormones are going to elicit some functionality within the cell to do a specific activity. So that's what we want to see. Is this is the cell wall, and those are the communication networks set up with the lock and keys represented in the green. So let's define again what a phospholipid is. It's a natural self-assembly lipid unit. The phospholipid is a molecule related to fat. The molecule is comprised of a hydrophilic water-loving phosphate head and two hydrophobic water-fearing fatty acid tail. When the phospholipids are added to water, they self-assemble. The phospholipid head points towards the water, keeping the fatty acid tails away from the water, forming a bilayer. And these bilayers, for us, could act as a cell wall or a cell membrane. Actually, cell walls are more for plants. I should have in the presentation membranes is probably more appropriate, so I apologize for that. So this is a phospholipid structure. We can see a phosphate head and then the fatty acid tails. And then often these are coupled together within the membrane. If I can go back up in the picture, these membranes here, the fatty acid tails, they could actually be cross-linked and made to be more durable, the wall, with proteins. So they have certain proteins that are characteristic, within, per se, like eggs, and we want to look at those as a cross-linking matrix within that cell membrane. So the nucleus itself is another form of the phospholipid biwall. It's also formed from the bilayer. It's the most prominent organelle in the animal cell. And what the nucleus does is it houses the DNA. And the DNA is responsible for protein synthesis through its helper RNA. So what happens is DNA has all the protein sequences that the body needs to do, and the RNA is a specific subsection of that DNA, and it carries that outside of the nucleus, and it would make the proteins at that point. So the nuclear envelope itself, made out of these phospholipids, protects the DNA. The DNA, like I said before, acts as the hard drive, and it contains all the programs the body needs to carry out life functions. And in some senses, by analogy, the programs would be specific proteins. So again, as a review, we'll take a look at this generic animal cell here in the presentation. So we have this DNA strand, and what it's going to do is we can make a copy of that called RNA, export the RNA within the cell, and what will happen is the ribosome itself can ride along these. They look like railroad tracks, those little data points in there, and they're actually like a memory system. Three of those chemically would represent one amino acid. So three of these rails are going to be encoded, and each one acts as like a word. So it's like an ASCII system. Certainly it has nothing to do with IEEE or electronics, but it acts like a communication system, like a hexadecimal type thing, that these three chemical affinities would actually represent certain amino acids, and they would call that pair of three a codon. So the nuclear envelope itself, it's separated by the cytoplasm from the rest of the cell by the nuclear envelope. It's a double membrane bilipid layer separated by a space of 20 to 30 nanometers. The envelope is perforated by pores that are about 100 nanometers in diameter, and these pores regulate the transport of macromolecules and particles. And like I said before, these act as like a doorway, like a garage door, and they are on the order of about 100 nanometers. They could be bigger or smaller, but again, as a good rough number, that's important. So naturally, for the nanotechnologists, these size parameters determine the material interaction and material process tool set, and certainly the characterization tools. So for example, if we wanted to put a drug inside a cell membrane, probably the size of the drug being under 100 nanometers would be really efficient, because the cell would naturally not like to filter that. If we tried to make a 500 nanometer diameter drug and import it into the cell, that would be very, very hard to carry out, because the cell just doesn't like materials on that scale. So we would like to make things at 100 nanometers. And it's exciting to be an engineer at these times, because these are the tools that we have available for us. So the nucleus itself, as we said before, houses the DNA. And this DNA is also repackaged and reassembled as chromosomes during cellular division. So that's a key point that we'll look at later, because we want to tie up the DNA so that it can't be used during cell division. That way that we can stop cancer. So why do we care about DNA and proteins? Because DNA is an excellent example of controlled self-assembly. DNA is transcribed into RNA, so transcribed is the same per se language. It's still nucleic acid to nucleic acid, but then it is translated into amino acids, which is a different type of acid. So that's why they have the terminology transcribed and translated. So DNA is nucleic acid to a subnucleic acid, and then that system is actually used to create a amino acid, and this amino acid in its assembly form is called a protein. So the DNA is the software template, and then proteins themselves carry out the functions. So we're going to look at DNA and its activity first within the body, and see how it's going to be used to create proteins later in the presentation. So DNA itself, DNA consists of a series of nucleotides. These are the building blocks of nucleic acids, and they're made up of 5 carbon sugar deoxyribose covalently bonded to a nitrogenous base pairs and a phosphate group. The phosphate of one nucleotide is bonded to the sugar of the next nucleotide in line, resulting in a sugar phosphate backbone from which the bases project. As a definition, a codon is a set of 3 consecutive nucleotides in a strand of DNA or RNA, and this provides the genetic information to code a specific amino acid that will be incorporated into a protein chain to serve as a termination signal. So we're looking at a DNA structure. So this has 2 sides to it. So this spiral, whoops, hit the mouse there. It has 2 spirals in there. And interestingly, these spirals in some senses are a mirror image of each other. So anytime that we have a G on one side, we would have a corollary to its mirror side, a C. And anytime that we have a T on one side, that's actually always going to mate to an A. So basically, if we know one side of DNA, we would chemically be able to ascertain the other side of the DNA. And not only is that a mental exercise, this is how the body works. So during cellular division, what we're going to do is split the DNA and then reassemble the DNA into 2 clones. So we'll clone the DNA from one parent, split it into halves, and then we complete those halves into 2 wholes once again. So what the point here is one side would be able to tell you what the other side is. And basically that's a key of cellular division, is that we can take this DNA information and divide it between two cells, because we're going to be able to recreate that DNA in this cloning sequence. So this shows us a single strand of DNA. This is the covalently bonded sugar phosphate backbone, and that's really tremendously coupled together by high energy per se, and rather robust. So we know that they find DNA, hundreds or thousands of years old, and they're able to interrogate it. And this is on the chain of the DNA, like the T, A, G, and C, and these are going to represent, the specific nucleic acids at that point. And if we would consider three of these, like TAC, we would say that that is a codon. And then the next three, G, whatever it is, X, CC or whatever, then that would be the next codon. So these, like three of these terminal spots, are actually going to be encoded by the body to give us a specific amino acids, which is going to be integrated into a protein. So the roles of DNA, pyridines always bond to pyrimidines and vice versa, and never to each other. So again, bonding always occurs in the following convention: A bonds to T and G to C, and likewise T to A and G to C. So this allows half of the DNA chain to be replicated. So interestingly, the DNA in the human contains about 6 billion base pairs. Nature uses 3 base pairs to define a specific amino acid, and this is defined as a codon. So this is a table of codons for amino acids. So if we would have a certain amino acid given, GCC, then it would be this alanine right here, ALA. And so these are the different amino acids that are in the body, and this is the encoding that would be necessary to get those. So this is the first base of the codon, the second base, and the third base of the codon, and how these things would be arranged. And this shows the process of having codons, and the codon representation would give us polypeptides. And now once the polypeptide is assembled, the amino acids, when it's placed in water and it's able to form in its hydrophobic formation, it forms a three-dimensional structure, and this three-dimensional structure is actually an operational protein that could elicit responses within the body. So now we're going to take a look at the DNA structure. It's a double helix. We'll look at the scale and then the native form of DNA. It consists of 2 adjacent strands held together by hydrogen bonds. And we know from our look at through this course that hydrogen bonds are moderately weak. So that's the point where we're going to unzip the DNA. The double helix is about 2 nanometers in diameter. The base pairs are about 0.34 nanometers apart, and there are about 10 base pairs per turn in the helix. So this again shows us the DNA and some scaling on this DNA, which is rather important. So DNA self-assembly overview. DNA is an example of a controlled self-assembly. During cell division, DNA is split between the old and new cell. Before replication, the parent cell contains 2 complementary strands of DNA. Each parent cell's 2 strands are separated. The old strand serves as a template that controls the synthesis of the new complementary strand. So each DNA molecule consists of 1 old strand and 1 new strand. So later in this presentation, we'll look at nanotechnology that inhibits this DNA replication as a means to destroy tumors, and this drug is referred to as Doxil. So this is the DNA split during cellular division. So this is the cellular functionality of the cellular life. So basically what we're going to do during this transition, we're going to tie the DNA together so that we don't split the chromosomes apart into twins. So basically this is how we're going to cut the cellular division down is we're not going to allow this cycle to take place, because what we're going to do is tie the DNA together so that it can't separate to make 2 different cells. So this is a cellular DNA replication. So again, I referred to this before. For every G, you get a C. For every A, you get a T. So when we split the cell, what we're going to be able to do within the body is recreate the original, and then we're going to have 2 cells that were exactly looking like the first cell. So this DNA split allows the cellular division. So this is what we're going to do is disrupt this life cycle of the cell. So cancer is known to have an uncontrolled rapid cell division. One way to stop cancer is preventing cell division. The man-made nanoparticle that we're headed to discussing is called the liposome, and in particular, the liposome we're looking at contains a drug that prevents DNA replication and therefore stops cell division. So this method of holding the DNA together is called intercalation, and that's what we're going to look at later in the presentation. So the DNA intercalators are used in chemotherapeutic treatment to inhibit DNA replication, and this is what they're used for, and this is what happens. So the DNA is like this, and basically what we're going to do is we tie the DNA just like we put a tie wrap on there or a chain, and now it's not able to divide at all. So that's our goal. Just our goal here in the discussion to look at the liposome. We really want to look at the manufacturing process of the liposome, but I think this is a good background and cellular function to know why we want to make liposomes or why they're so effective, or how we're following the boundary values of biology to make our man-made drug. So with that, I think it's time to stop. We're going to call this part 1. And we're going to stop right here, and then we're going to continue this discussion the next time and take a more detailed look at biology leading to liposomes. So thanks for your attention, and we'll see you next time. Take care.